Have you ever imagined a world where we can rewrite our own DNA to cure diseases? Well, that world is arriving faster than you think. The field of genetics is exploding with breakthroughs that sound like science fiction but are quickly becoming reality. From personalized medicine to AI-designed DNA, we're on the verge of a revolution. Today, we're diving deep into the most incredible genetic advances happening right now. Stick around, because what you're about to hear will change how you see the future of humanity. Let's start with a story that's both heartwarming and scientifically groundbreaking. Earlier this year, a 9-month-old baby with a rare and life-threatening metabolic disorder became the first person ever to receive a truly personalized gene editing therapy. This wasn't a one-size-fits-all treatment. Scientists designed a unique CRISPR-based therapy specifically for this child, targeting the single faulty letter in their DNA that was causing the disease. Think about that for a moment. Instead of just managing symptoms, doctors went straight to the source code of the illness and corrected it. They used a technology called a base editor, which is like a hyper-precise pencil and eraser for our genes. It can change a single DNA letter without making risky cuts across the entire DNA strand. This incredible success story isn't just a win for one family. It's a beacon of hope for thousands of others with ultra-rare genetic conditions. It signals a new era of bespoke medicine where treatments are tailor-made for the individual. While CRISPR has been the star of the gene editing world for a while, it's no longer the only tool in the box. Scientists are constantly discovering new systems in nature that can be harnessed for genetic engineering. One of the most exciting new players is a system called TIG-ARTAS, discovered in bacteria and their viruses. TIG-ARTAS works differently from the classic CRISPR-Cas9. It uses to guide RNAs instead of one, which work together to cut DNA. So, why is this a big deal? Well, the classic CRISPR system needs to find a specific short sequence next to the target gene, called a PAM site, to work. This can sometimes limit where we can edit. TIG-ARTAS doesn't seem to need this PAM site, which could give scientists much more flexibility and precision. It's like upgrading from a tool that only works on certain types of screws to a universal wrench that can tackle any job. More tools mean more possibilities and a wider range of diseases we can potentially target. The next frontier is not just about editing existing genes, but about designing entirely new genetic instructions from scratch. And for that, scientists are turning to a powerful ally: artificial intelligence. Imagine you want to turn on a gene, but only in a specific type of cell, say a liver cell, and not in a heart cell. That's an incredibly complex biological challenge. But now, a new AI framework called CTRL-DNA is making it possible. It uses a type of AI called reinforcement learning. The same kind of tech used to train AI is to master complex games, to design brand new DNA sequences. These AI-generated sequences act like highly specific on switches or promoters for genes. They can activate a gene precisely in the target cell type while staying switched off everywhere else. This level of control is a game changer for gene therapy. It could help make treatments safer and more effective by ensuring the therapeutic gene only works where it is needed, minimizing side effects. The synergy between AI and genetics doesn't stop there. One of the biggest challenges in biology is understanding how genes work together. Often, tackling a complex disease requires targeting not one, but multiple genes. But with thousands of genes in our genome, testing every possible combination is practically impossible. It would take decades and millions of experiments. Enter NAIAD, another brilliant AI-powered method. NAIAD uses a clever approach called active learning. It starts by running a small number of CRISPR experiments, then uses that initial data to predict which other gene combinations are most likely to have a powerful effect. The AI essentially tells the scientists, based on what we've learned so far, you should test this pair next. This massively speeds up the discovery process. Instead of searching in the dark, researchers can use AI as a flashlight to find potent gene pairs that could lead to powerful new combination therapies for cancer and other complex diseases. These incredible advances in the lab are exciting, but what matters most is whether they can actually help people. The good news is the pipeline from scientific discovery to clinical application is getting shorter and shorter. In 2025, we're seeing a surge in clinical trials for rare genetic diseases using the very latest gene editing tools. And regulators are taking notice. This year, the FDA approved a new gene therapy called ZEVASKIN. It's designed to treat a devastating rare skin disorder that makes skin incredibly fragile. This approval is a huge milestone. It shows that gene therapies are not just a theoretical promise anymore. They are becoming a standard part of our medical toolkit. Every approval paves the way for more treatments to follow, bringing hope to patients and families who previously had none. Of course, with great power comes great responsibility. The ability to easily write and rewrite DNA is one of the most powerful technologies humanity has ever developed. And as it becomes more accessible, we have to think seriously about safety and security. How do we prevent this technology from being misused? Scientists are already working on solutions. One ingenious idea is to create watermarks for synthetic DNA, just like an artist signs their painting. Biologists can embed a unique, hidden signature within the DNA sequences they create. This watermark is designed to be passed on even when the DNA is translated into a protein, making it possible to trace the origin of any engineered biological material. It's a crucial step toward creating a framework for responsible innovation, ensuring that we can track and verify the source of synthetic DNA and promoting transparency in the field. Beyond just editing genes, scientists are also building with them. This is the realm of synthetic biology, where DNA is used as a building block to create entirely new biological systems. Imagine creating tiny, self-assembling biological machines that can deliver drugs directly to a tumor or smart materials that can repair themselves. This is exactly what researchers are doing. They are engineering synthetic gene networks that mimic natural processes to build complex, nanoscale structures. These genes can be programmed to produce proteins that automatically link together, forming new biomaterials with incredible properties. This could lead to advanced medical implants, highly sensitive biosensors, or even smart therapies that can sense their environment and act accordingly. We are literally learning to program life itself. As we look at the bigger picture, it's clear that the future of genetics is deeply intertwined with artificial intelligence. This isn't just a temporary trend, it's a fundamental shift in how we do biology. AI models are now indispensable. They help us predict the potential side effects of gene editing before we even do an experiment. They design the most efficient guide RNAs to ensure CRISPR works perfectly. And they help us make sense of the mountains of data generated by modern genomics. For instance, researchers are now combining large-scale CRISPR screens with single-cell analysis. This allows them to see, with unprecedented detail, exactly how knocking out a specific gene affects every single cell in a population. It's like going from a blurry, black and white photo of a city to a high-definition 4K drone video where you can zoom in on every single person. This level of resolution is unlocking fundamental new insights into health and disease. So, where does this all leave us? We're witnessing a true paradigm shift. From the first personalized CRISPR therapy healing a baby, to the discovery of new editing systems like TIG-ATAs and the use of AI to design and understand our own DNA. These aren't just small steps forward, they are giant leaps that are reshaping medicine and biology. At the same time, it's reassuring to see the scientific community being so proactive about the ethical and safety challenges. By developing tools like DNA watermarks, they are building the guardrails needed to ensure this powerful technology is used for good. Together, these innovations are building a future where genetic diseases are no longer a life sentence, where we can design biological systems to solve our biggest problems, and where the incredible power of genetics is wielded safely and responsibly. The future is being written in our DNA, and it looks brighter than ever. Thanks for watching. If you found this as fascinating as I did, do not forget to like this video and subscribe to the channel for more deep dives into the science that is shaping our world. See you next time.